I'm speaking to you from my virtual office at MIT. I am so honored to be receiving an award named for Pat Goldman-Rakic. Pat was an inspiration throughout my career, and her two major areas of research have been the role of prefrontal neural circuits in cognitive function, and then how dysfunction of these circuits may lead to a variety of brain disorders. And those are my interests as well, and I'm going to be touching on both of these topics during my talk today. One of the areas of cognition that I've been focused on for the last several years is on the neural basis of attention, how we pay attention to things. And that involves a visual pathway in the primate that splits up into two major directions according to this anatomical diagram from Leslie Ungerleider. One is directed from the primary visual cortex, or V1, up into the parietal lobe, and is comprised of these areas shown in green. And this is this pathway that's important for spatial perception. And the other is directed down into the temporal lobe. It's comprised of these areas shown in red, and is important for object perception. And then there are extensions of both pathways up into the prefrontal cortex. Now, the pathway that I've been focused on is the one for object recognition, both how this pathway ends up recognizing objects, but then also how it is that when we pay attention to things, this greatly modulates how we process visual information in this pathway. How does that come about? Now, we know from work in both my own lab and many other labs that as you move along this pathway, the receptive fields of cells, it's the parts of the space analyzed by cells, increases. So from in the primary visual cortex, you have pixel-based receptive fields, and then they get bigger and bigger and bigger as you move along. And by the time you get to the inferior temporal cortex, you could have receptive fields that include these entire scenes. So there's a tremendous amount of clutter in the receptive fields. However, we know from recording along this stream that there's a progressive enhancement of processing of behaviorally important objects, the one we're attending to, and then suppression of objects that are distractors. So that by the time you get to the inferior temporal cortex, you could have just a representation of the relevant object. So for the student, it's the book in front of her that she's paying attention to. Now, I mentioned that we see along this pathway this enhancement of processing for relevant objects. And this is just an example here from a population of cells in area V4, which is midway along that processing stream. Collected when animals were trained to fixate a spot, and then we could place a stimulus in the receptive field of a cell, like you see here, and then another stimulus outside, and we could direct the animal's attention to one or the other stimuli. And when we look at the firing rate histogram, what we see is that when the animal pays attention to the stimulus, there's this enhanced response. And when the animal ignores the stimulus, there's this more suppressed response. And at the same time, there's not only these changes in firing rates, but there's this increase in what we call spike field coherence. This is how well the cells are synchronizing their activity with each other. And what we see is when the animal pays attention to the stimulus, there's this enhancement of synchrony in the gamma frequency range. Now we know from lots of other experiments that the combination of these two effects, the increase in firing rate and this increase in gamma frequency coherence, are going to have a big impact on the next area downstream in this pathway, and it will lead to this enhanced processing and suppression of distractors in the next area. And this goes on from stage to stage along the pathway. Now, how does this come about? Where is the information coming from that's modulating responses according to what the animal's paying attention to? Well, from work in our own and other labs, we know that there are these systems for the top-down control of attention, which are present in both the parietal cortex and the prefrontal cortex. And today I'm going to focus on the prefrontal components of this, and particularly an area in the prefrontal cortex known as the frontal eye fields, and its role in in providing this feedback to the ventral stream areas. If we record simultaneously in the frontal eye fields in this intermediate area that I've already mentioned, area V4, and in the same kind of task where the animal is paying attention to a stimulus or ignoring it, and we look at the population response, we can see that in both areas there is an enhancement of the response when the animal pays attention to the stimulus, and this greatly reduced response when the animal is ignoring exactly the same stimulus. As far as the retina is concerned, there's no change. And if we look at the timing of these effects, though, what they tell us is they're not identical across the two areas. The effects are starting earlier in the frontal eye fields at 80 milliseconds compared to V4, which they occur later. And this suggests that the frontal eye fields is the cause of the effects that we see later in V4. Likewise, if we look at coherent interactions between these two areas in this gamma frequency range, the cells across the two areas are synchronizing their activity when you're paying attention to a stimulus. It's greatly enhanced with attention, and we've shown through other analyses that it's the frontal eye fields that is driving V4 in these interactions. Furthermore, in the experiments I'm not going to show, if we create a lesion in the prefrontal cortex that includes the frontal eye fields, these attentional effects in V4 are reduced. So it's all evidence for a causal role in the frontal eye fields in modulating our visual system by attention. But this is all, as I mentioned, spatially directed attention. But we know there are other kinds of attention, in particular paying attention to objects. And to give you an example, if I ask you to look at the scene and to try to find the girl in the pink shirt, I'm sure you can do that. And yet it's really perplexing how it is that you do it, because we know that our brain contains multiple maps of the visual field, including in the attentional system. And so if I were to tell you, say, to attend to the upper left-hand corner of the screen, you could do that, but it would be associated with increased activity in one of these maps in the upper left-hand corner. So that's pretty straightforward. But when I ask you to attend to the girl in the pink shirt, like, where's the map of the girls in pink shirts? Where's that come from? That seems much more complicated. And in fact, we think it's very deeply related to a similar situation when you recall items from memory and hold them in your working memory. So for example, if I asked you to remember what your mother looks like and hold her image in your mind's eye, probably most of you could do that. And we know from experiments that have been done in human imaging that when people are imagining things from memory, this kind of loads up the areas in the visual processing system as if they're holding that image. This is a problem that Pat Goldman-Rakic was very interested in, is how do we maintain objects in memory, in working memory? How does this turn about? And we think it's very deeply related to this problem of how do we find objects based on attention to them coming up from our memories? So I'm not going to go through the details of the experiment, but we did an experiment in human subjects for this object-based attention. And in this case, we had the subjects attending to either faces or houses that were spatially superimposed. So there's no spatial difference between them. They're on top of each other. And what we found was that when they were attending to either faces or houses, there was this region in the prefrontal cortex known as the inferior frontal junction, or IFJ, that was active in both the tasks. However, when the subjects were attending to faces, we saw synchronous activity with an area in the ventral stream that's known as the fusiform face area that's important for processing faces. Whereas when we asked them to attend to houses, then we saw the synchronous activity with the parahippocampal place area, which is known to be involved in the perception of things like houses. And so the IFJ is sort of switching back and forth depending on what kinds of objects you're attending to. Just like the frontal eye fields is sort of switching back and forth depending on where in space you're attending to. In fact, we see very parallel systems between the frontal eye fields visual cortex system that we see in primates and the IFJ object processing systems that we see in humans. So how could we use the primate experiments to better understand how this comes about is there an area in primates that corresponds? So in here, what we did was record it from the frontal eye fields and simultaneously from an area that we think is analogous to the IFJ in humans. We called it the ventral prearcuate area in monkeys, which is just in front of it and ventral to it. And then we recorded from ventral stream areas as well. And the task that we used was very similar to that task I showed you where I asked you to find the girl in the pink shirt. In this case, the monkeys are fixating, and then we show them a cue object. Let's say it's a shoe in this case. It changes from trial to trial. And then there's a blank fixation period. And then on the search trials, an array of objects appear on the screen. And the monkey's task is simply to move its eyes around until it finds the object that matches the cue. And we call that the target. So it finds the shoe in the array, and it just holds fixation there. Okay, so we have the monkey doing this task. How do we separate out the signals for spatial-based attention versus this attention to objects? Well, for spatial-based attention, which is shown down below, it's very simple. We either have the monkey make an eye movement and attend to a stimulus in the receptive field of the cell we're recording from, or the monkey makes an eye movement someplace else and is tending to something else spatially. So we can just compare the responses in those two situations. And that gives us this enhancement by attending to things. And whereas in the spatial case, the feature case, we look just at those intervals where the animal is about to make an eye movement to something else, not in the receptive field. But in some cases, the thing that's in the receptive field is the thing that the animal's searching for. And in other cases, it is not. So we look at the response, say, to the shoe when the animal is looking for a shoe, and we compare that to the response to the shoe when the animal is looking for a banana, say. Okay, so now we have ways of separating out these signals. What are the results? Well, up top, we're showing the task conditions, both the cue and the delay and the search array. And down below, we have the population response of these neurons. Now, the neurons in this VPA area show selectivity for objects, just like we see in the temporal cortex. Some cells respond better, let's say, to a shoe, other to a face, other to a house. Whatever it is, they have this kind of selectivity. And we see a larger response for their, what we call the preferred stimulus for a cell compared to a non-preferred. So they're carrying information about the cue. But now, interestingly, if you look during the delay period, now this is a period where the animal's just looking at a blank screen, waiting for the array. But during this memory delay period, you can see that the activity is higher if the animal is going to be looking for the stimulus that was the preferred stimulus for the cell compared to a non-preferred. And this delay activity, Goldman-Rakic also studied throughout her career, and she referred to as the memory-related activity, working memory-related activity. And we see it as well. And then when the array comes on, you can see that for the cells that have this higher activity for the preferred stimulus, the response rate is already higher. It's as though your circuits in prefrontal cortex are primed to detect that object in the array. Now let's look at what happens when you when the animal's searching around the array. Now here we're looking at the activity in the frontal eye fields and the VPA. And what we have is the population responses again. And without going through the details of this, the green line shows the population response when the animal's paying spatial attention to the stimulus versus the red line. And so that difference between the red line and the green line is a measure of the enhancement by tending to that location. And whereas the difference between the red line and the blue line is the enhancement that you get from attending to the feature in the receptive field. So those are the two signals we're looking for, and they're in the frontal eye fields. If we look in VPA, we see exactly the same two signals related to the spatial enhancement and feature attention enhancement. However, if we look at the timing of these signals, the feature signal is present earlier in VPA, and the spatial signal is present earlier in the frontal eye fields. suggesting that maybe the VPA is supplying this information to the frontal eye field so that it can cause you to direct your eyes to the stimulus. But then we need a causal experiment to test that. So what we do is we deactivate the VPA in one hemisphere with mucimol, which just turns off all the activity. And then we measure the animal's performance when it's doing this task, just behavioral performance at first. And we look at both the ipsilateral and contralateral visual field, and both prior to the injection of mucimol and after. And what you see is if you look in the ipsilateral visual field, which is unaffected by this deactivation of EPA in one hemisphere, there's no difference in performance. But in the contralateral visual field, which is getting that hemisphere is getting the feedback from the VPA that we've deactivated, now you see that the animal's making many more errors with that VPA deactivation. And we see sort of an intermediate effect in the midline. So this tells us that VPA is actually needed to do this task correctly. And now let's look at what happens in the frontal eye fields. Well, if we look in the frontal eye fields before the deactivation of the VPA, we see these two signals that we've been searching for, the spatial attention enhancement and the feature attention enhancement. So that's just what I showed you previously. But if we record after we've deactivated the VPA, the enhancement for the feature attention is gone, but the enhancement for the spatial attention is totally normal. So what this tells us is that when we've deactivated VPA, we've eliminated the source of information that's going to the frontal eye fields, telling the frontal eye field cells where is the object that the animal's trying to find right then, whether it's the shoe or the banana or whatever. It loses that information. The frontal eye field only knows where you're going to move your eyes, and you're moving them often in error. So this, again, is strong causal evidence for the role of VPA in this feature attention. In fact, what we think is that the frontal eye fields and VPA are working together, with the VPA identifying the object you're trying to find, it's holding this in your working memory and supplying that information to the frontal eye fields that can direct your eyes to it. And both of these areas are sending feedback to this ventral processing system. And I'm not going to have time to show you the experiments, but if you deactivate either the frontal eye fields or VPA, you see this reduction in the attentional modulation of cells in this pathway, as you'd expect. So we think we have the causal role of these areas. Now, lastly, I just want to touch on this other topic that occupied a lot of Pat Goldman-Rakic's research, which is the role of prefrontal and other cognitive mechanisms in brain disorders. And her particular interest was in primates, as is ours as well. And one of the reasons for this is that whereas work in mice has been absolutely essential to our understanding of the biology of the brain and neural circuits and even the neural basis of brain disease. But the problem is that when treatments have been developed in mice, they often don't translate into clinical success in humans. You find a drug or whatever, or another manipulation that seems to work in a mouse, you try it in people, it doesn't work, or it has terrible side effects, and so on. So, one possibility, of course, is that the mouse brain is very different from the human brain. Of course, it's much smaller. But there's also many structural differences in terms of number of neurons and packing of neurons and so on, organization of neurons, even sites of cells. Whereas the non-human primate brain is intermediate and has many more similarities to the human brain, which, of course, a human is another primate. And if you look at the medial prefrontal cortex in particular, which is a region to be known to be very important, for example, in social cognition, it's very large and elaborated in humans, comparably elaborated in primates, and very small and almost rudimentary in mice. So given that, it seems like we have, if we could develop these animal models of disease in primates, we would have a much better chance of developing treatments that could translate to humans. Now, fortunately, we now have that capability with the CRISPR method for creating genetic-based disorders. And in an experiment that we've done now in collaboration with our colleagues at the Shenzhen Institute of Advanced Technology in China. Guoping Fang, who's been the leader on the US side of this collaboration, and Zhonghua Lu, who's been the leader on the Chinese side, have created a model of a disorder in humans that's a subtype of autism, and it's caused by a mutation in the SHANK3 gene. It's known as the Phelan-McDermid syndrome. And using the CRISPR method, you can inject the embryo with a CRISPR construct, cause the mutation, then the embryo gets transferred to a surrogate mother, and the offspring then can carry this mutation. And then we can study those animals as a first step to see if it's a good model. And when we've done that with the SHANK3 mutation, what we find is a number of behavioral impairments that seem reminiscent of this form of autism, which is a very severe form. I mean, this causes many of the people with Phelan-McDermid syndrome are institutionalized. And one thing we see is reduced vocalization. It could be related to the fact that many people with Phelan-McDermid syndrome actually don't have speech. We see this reduction in social behaviors. And if we pair the animals with a normal probe animal, what we find is even the probe animal has less social behavior when they're paired with an animal with this mutation, which is, I think, interesting. If we test them in a task, the set-breaking task, which is known to be sensitive to prefrontal function, we see that the animals with the mutation are impaired. And if we measure the functional connectivity of their brain regions using MRI, we find that in a region of medial prefrontal cortex, that region I was just telling you about, this is the site 3 here, that there's reduced global connectivity with the rest of the brain in these animals with the mutation compared to the normal controls. Again, it's a very striking parallel to what we see in the syndrome. So this is giving us great encouragement now that we can now develop treatments which include gene therapy and medications for these animals that could hopefully correct some of these problems and then would lead to human clinical trials that we hope would have a greater probability of success. And we're now expanding this to other genetic models and brain disorders as well. And all I can say is, I think if Pat Goldman-Rakic were alive, she'd be jumping all over these methods, and she'd be wanting to create these genetic models too. I think she would be very pleased with how the field of cognitive neuroscience is progressing, and that we may actually be able to achieve our goal of being able to help people with some serious disorders. Thank you very much for your attention.